Hello, today we are investigating the effect of salt on the bacterial growth of kimchi. People should always take note of the amount of salt they use in the process of making kimchi, especially if they want the fermentation process to last longer. Even after extensive research, there's no results on the ultimate amount of salt to make kimchi that will last the longest. Hence, we decided to take it up on our part to find out what would be the best amount of salt for making kimchi.
The people turn to fermentation to preserve it. Fermentation is the chemical breakdown of a substance by bacteria, yeast, or other microorganisms, typically involving effervescence. Effervescence is the bubbles in a liquid or otherwise known as fizz. In this experiment, we wanted to find the effect of salt on the bacterial count of kimchi. To achieve this goal, we added a varying amount of salt to standardize the amount of kimchi. The amounts are increasing orders of 5 grams of salt, with 0 grams, 5 grams, 10 grams, 15 grams, 20 grams, and lastly 25 grams. Our hypothesis was that the higher the amount of salt added, the lower the percentage of bacterial growth on the agar plates we used. First, we made kimchi using cabbage, water, all-purpose flour, brown sugar, radish, carrot, spring onions, chives, garlic, ginger, onion, fish sauce, and chili flakes. Then we separated it into 6 containers and 100 grams of kimchi per container. We then added salt accordingly to the sequence mentioned just now. The increasing order of 5 grams of salt with 0 grams, 5 grams, 10 grams, 15 grams, 20 grams, and 25 grams. We then brought it to school to begin our experiment. We began by swabbing a 2 cm by 2 cm square on the kimchi and washing it in saline solution. Then, using a micropipette, we extracted 100 milliliters of solution out of the tube, then ejected it onto the agar dish. Using an L spreader, we spread the solution equally onto the surface of the agar dish. After that, we covered the agar dish with the petri dish cover and labeled it by grams, class, group, and date. We repeated the same procedure for the other agar plates with the kimchi with the different amount of salt. As we were doing 3 batches, we repeated the experiment 2 more times. After which, the agar plates were placed into an incubator for incubation for a total of 11 hours. For the data analysis of our results, we made a 10 by 10 grid in Keynote, then took a square screenshot of the photo we had taken of the bacteria and imported it into the grid. We roughly estimated the percentage of the surface area of the bacteria. We compared the data and took the average of the data as our main data. We found the average percentage out of the 3 setups and made graphs of our result. From our results, the kimchi with the second most amount of salt, which is 20 grams, had the lowest amount of average area percentage and the highest being the kimchi with the least amount of salt, which is 0 grams. The second highest being 10 grams, followed by 5 grams, 15 grams, and lastly 20 grams. The results could be due to the salt drawing out the water from the kimchi cells and bacteria through osmosis. When the amount of water in the cells are reduced, bacterial growth stops or slows down. This means that our hypothesis was correct. Fewer bacteria can grow in the presence of salt. The more salt there is, the smaller the surface area of the bacteria grown. Salt has a big part in stopping the bacterial growth in food during fermentation. The most notable is simple osmosis or dehydration, thus killing the bacteria. By sharing this information, people will be able to take note of the amount of salt they use. If they want it to ferment longer, they can add more salt. This is especially important in hot climates or when storing food without refrigeration.
In our experiment, we had a lot of ups and downs with the results and analysis, especially with getting the results of the bacteria area of each agar plate. We definitely learned a lot from this project, like how to conduct a research project. We also learned how to grow bacteria, which was a very interesting and fun experience. We would definitely want to try it again, as we got to use many different equipment that we usually won't use. Overall, while we faced many challenges, we still managed to pull through and had many great learning experiences along the way. That's all for today. Thank you.